everyone. Thank you so much for the invitation to speak today. Again, my name is Mary Frances Wedekind, and I am a pediatric oncologist in the pediatric oncology branch in NCI at NIH, and I am the principal investigator of the natural history study of rare solid tumors in pediatrics and adults. And I am very happy to present today an overview of the Cancer Moonshot-funded My Pediatric and Adult Rare Tumor Network, or MyPART. Rare tumor definitions vary across multiple sources, but for our group, we utilize that a rare tumor is an incident of less than 150 cases per million per year, thus making up about 27% of newly diagnosed cancer. While all pediatric cancers technically fall under this definition, you can also look at very rare pediatric cancers, which are an annual incident of less than 2 per million per year, making up of only 11% of pediatric cancers. There are numerous challenges within rare tumors, from the difficulty in establishing the diagnosis, no standard of care with overall incomplete understanding of the natural history, and difficulty conducting disease-specific clinical trials. Overall, rare tumors across the world are lacking in adequate knowledge, resources, and community. MyPART was developed in support of Cancer Moonshot funding to engage all of those involved in rare tumors, from patients, family members, and advocacy groups to researchers and healthcare providers, to partner to improve the lives of young people with rare cancers. Through multi-institutional collaboration, we developed a natural history study, hold workshops, and host multi-day specialty clinics with a goal to collect, analyze, and share all available data to develop an increased understanding of tumor biology and natural history, which will then be translated into new clinical trials. The natural history study of rare solid tumors in pediatrics and adults uses standardized retrospective and prospective comprehensive longitudinal evaluations of patients with rare solid tumors and their family members, being led by myself and Heidi Del Rivero, who is an endocrinologist and medical oncologist that focuses on endocrine cancers. Children and adults at any stage of their cancer journey, including those newly diagnosed or with relapse refractory rare solid tumors, as well as their biological relatives, are invited to join. Participation can occur at the NIH or offsite anywhere in the world, including their own home. After enrollment, patients or their family members on the protocol electronically complete standardized medical and family history forms and patient-reported outcomes. With pathology specimens from all patients with a rare tumor received, we are able to complete molecular profiling with results discussed at a multidisciplinary review and then return to the patients with the help of genetic counseling. All patients are given a medical opinion, including clinical trials, not only at the NIH, but also across the country or internationally if needed. If applicable, patients have the ability to participate in sub-protocols where more disease-specific questions are asked. For those who come to the NIH, imaging, biopsies, surgeries may be obtained if clinically indicated. For participants with a rare solid tumor or those without a tumor but found to have a germline mutation, we follow up yearly for updated questionnaires, medical record and biospecimen collection, and provide new treatment recommendations. None of the evaluations performed on this study are billed to the participant or insurance, with funding also to support travel and lodging. For all participants on the natural history study, we collect medical records throughout the patient's cancer journey and extract data on standardized collection forms, including data such as presentation, treatments, current disease status, and pathology elements. We also collect tumor tissue from as many different time points as feasible. The questionnaires completed by participants include medical and family history and PROs. For participants with the opportunity to come to the NIH, we are able to perform further clinical data with blood draws, physical exams, imaging or biopsies as medically necessary, with the development of cell lines and PDX models from collected tumor tissue if feasible. Our genetic counselor is also able to meet with our participants for further pedigree analysis and phenotyping. Seen here are the forms that are completed annually, with the forms in red, which includes all of the PROs, being completed by the participant. Forms in green are completed for all participants, and the forms in blue are completed for those participants that come to the clinical center. In addition to confirming diagnosis from tumor tissue, we look for actionable gene mutations followed by extensive research analysis. We collect saliva from all participants by sending kits to their home with return envelopes, which is then used for clinical and research analysis. For those who come to the NIH, blood samples are collected for detailed analysis to look for circulating immune cells and potential biomarkers of disease. Shown here are our lymphocyte and myeloid panels to evaluate circulating immune cells. And here is our cytokine panel utilizing the VPlex 36-plex kit. Since opening for enrollment in January of 2019 until May 2022, when these numbers were pulled, we have enrolled about 451 participants, which are adult and pediatric, and include 55 different histologies. We are currently almost up to 500 participants to this day. Enrollment spans 6 continents and 22 countries outside of the US, with most states within the US included. Seen here are the tumor types enrolled on our protocol, with the most common including NET, STH deficient or wild type GIST, ACC, chordoma, and medullary thyroid carcinoma. You can see in the blue which tumor types have a subprotocol attached to look for more disease-specific questions. Also, see the list of tumor types with only 1 or 2 participants. 8 participants have more than 1 tumor, which sometimes are not rare or solid, which is why they are listed in the N of 1. We have just started to look at some of our participants. So looking at our first 200 patients and evaluating the tumor samples, we attempted panel sequencing in 167 of the first 200 participants. We found that while age was important, other factors such as tumor density and tumor matrix also impacted the ability to collect DNA and RNA. Now looking at the sequencing results of the top 5 diagnoses out of our first 200 participants demonstrates that mutations were found in majority of the tumors. For panels A through F, red indicates pathogenic, yellow likely pathogenic, and blue is a VUS. ACC demonstrates the most variety in mutations, with TP53 being the most common. Chordoma showed the least mutations, with 2 patients with poorly differentiated chordoma harboring a mutation in this SMARCB1. In panel G, we highlight the highest actionable tier from 1A to 3 for the mutations in each participant. Not surprisingly, medullary thyroid carcinoma had the most evidence for targeted therapy, while chordoma and NET had the least. With all this data that is collected on participants, we return as many results as possible. NIH has a patient portal like many other institutions. We communicate treatment recommendations to the patients and their physicians. And following our weekly molecular tumor board, the genetic sequencing results are discussed to the patient with help from our genetic counselor and the treating clinician. Another aspect to MyPART is our outreach and communications. Through the support and educational resources on our website, our newsletter and Twitter account, we desire to enhance the experience to the patients to improve patient ownership. Patients want to see not only their own results, but the results of something that they are a part of. In addition to our patients, there are numerous benefits of having a strong partnership with advocacy organizations. To date, we have established 24 advocacy partnerships as listed here. We're very grateful for these partnerships and look forward to expanding them. Our successful enrollment of very rare tumors would not be successful without these partnerships. One unique aspect of our program that requires collaboration with patients and advocacy are our rare tumor specialty clinics. The wild type GIST clinic was started 12 years ago by Lee Hellman. During these clinics, 10 to 15 patients with a select very rare tumor are brought to the NIH along with multiple disease experts from across the country and at NIH. Robust data can be collected during this 3-day event. The wild type GIST clinic led to genomic characterization, increased clinical understanding, mouse models, and clinical trials. We now have specialty clinics for chordoma and MTC with the hopes to open other clinics in the future. One of the tumors of high interest to our program and myself is chordoma. Briefly, chordoma is a rare, slow-growing sarcoma originating from the notochord and occurring at the base of the skull along the spine and sacrum. There are only approximately 325 new cases a year, with only 5% of those being pediatric. Chordomas can be one of 4 histologies: conventional, the most common; chondroid, which is no longer in the HWO description; dedifferentiated; and poorly differentiated, which is more common in pediatrics. The distinction is important because this determines treatment, with conventional, chondroid, and dedifferentiated treated primarily with surgery and radiation, while poorly differentiated does not have a standard of care, but typically is first treated with chemotherapy, followed by local control, and sometimes immunotherapy. No matter the type, chordoma treatment requires multidisciplinary approach, including neurosurgeons, ENT, radiation oncologist, medical oncologist, and pediatric oncologist. Now to describe our success in chordoma. From a very high interest from numerous physicians and the establishment of a working group, a workshop was developed to include laboratory researchers, clinical researchers, advocacy, and clinicians as a starting point to collaboration. This led to the establishment of a partnership with the Chordoma Foundation, who then helped develop the chordoma sub-protocol on the natural history study, and then start the chordoma specialty clinic in 2019. With collaboration strengthened after the clinic, we now participate in multiple institution virtual tumor boards. And a very exciting opportunity that has come out of these collaborations is our pediatric chordoma working groups. Due to the rarity of pediatric chordoma, the natural history is not well understood. These working groups are critical to understanding the natural history of chordoma in pediatrics, as there are many disciplines involved and expertise is needed within all of these. We are utilizing these groups not only to develop data collection forums, but also in analyzing these results. Ultimately, our desire is to develop clinical trials for these patients. A brief look at our MyPART chordoma cohort includes mostly conventional chordoma that are in the clivus with a median age of 34 years. We have about 10% of poorly differentiated chordoma, which correlates to the SMARCB1 deficient tumor table seen here. Finally, through our tumor collection efforts, we have successfully made a chordoma PDX model. We currently are continuing patient enrollment with plans for detailed data analysis in the future and continue our collaborations with the Chordoma Foundation and numerous other researchers in the field, including the CBTN and Adam Resnick. As mentioned earlier, one of our major goals of the initiative is to develop clinical trials. A phase 1-2 trial of 2 checkpoint inhibitors for patients with SMARCB1 or SMARCA4 deficient tumors is now open through the Children's Oncology Group being led by myself and Vandana Ashkantala. Due to our foundation built through patient engagement and physician collaborators, we were able to justify a specific cohort for poorly differentiated chordoma despite its rarity. This trial is coordinated with CTEP and the PEP-CTN. Through the development of NIH rare tumor clinics, MyPART, in collaboration with advocacy and rare tumor experts, has a unique opportunity to gain insights on rare tumors. However, meaningful data collection is resource and time intensive, with a much more focused approach on select tumor types needed to ensure sufficient patient numbers. Partnership with all those involved in rare tumor research is critical to accelerate rare tumor progress. Yesterday in the CBTN meeting, Dr. Riemann spoke about the Childhood Cancer Data Initiative, and through these efforts, we are hoping to develop a collaborative national strategy for very rare pediatric and AYA cancers. In summary, MyPART has made substantial progress in engaging patients, researchers, and advocacy groups for the study of rare tumors. We've established a strong foundation for the collection and analysis of linked clinical and molecular data on rare tumors. Our studies have resulted in the development of new collaborative pediatric and adult interventional trials. MyPART is creating a critical infrastructure that is starting to be leveraged beyond its own efforts. I would like to thank the entire MyPART team for all of the hard work, NCI for the funding and support, the advocacy and research community for the collaboration, and lastly, the patients and families whom without none of this would be possible.